Good morning, everyone. I am Rayana Feltrin, and I'm currently a master's student in the Laboratory of Photobiology at the Federal University of Santa Maria, located in South Brazil. Today, I will be presenting to you my work entitled Occurrence of XPD helicase in Galliform birds. Well, genomes are constantly vulnerable to damage coming from different sources. However, several DNA repair mechanisms developed throughout the evolution of species to ensure the maintenance of genome integrity. The most versatile of them is the nucleotide excision repair, NER for short, as it can remove a wide variety of bulky lesions. As you can see here, the process of repair by NER occurs basically in 4 steps: damage recognition, excision of damaged nucleotides, DNA resynthesis, and strand ligation. The XPD helicase integrates the transcription factor 2H and has an important role before nucleotide excision in order to allow the separation of the double helix into single strands. XPD is one of the most conserved proteins of NER, being present even in archaea. However, a canonical XPD is apparently missing in Gallus, as we concluded in a previous study, so we decided to investigate further. In order to do so, we have carried out a refined search of XPD using sequences of the American alligator as queries for T-BLAST-N and BLAST-X searches against the genomes and proteomes of 15 species of galliforms, the order of chicken, in addition to 4 species of ctenomyforms and 1 of struthoniforms, which are in the base of the bird phylogeny. Then, we used 4 filtering criteria to select the ortholog sequence candidates: a value lower than 1e to the -6, highest percent identity, closest protein length to the query, and conserved protein domains, obtained by using SMART and PFAM. As the outcome, our search for XPD in chicken retrieved a sequence annotated as an ATP-dependent DNA helicase DDX11, which has the same XPD characteristic domains as you can see in XPD of humans. In fact, DDX11 is a member of the XPD family and acts as a backup for another kind of DNA repair in chicken cells. Therefore, we infer that DDX11 may have an XPD function in chicken, but functional studies are strongly required to test this hypothesis. Regarding other galliforms, we obtained some sequences annotated as hypothetical proteins in species such as Bambusicula thoracicus, Calipipla squamata, and Colinus virginianus, which contain the same XPD domains, indicating they may also have an XPD function. In addition, we retrieved some sequences that, spite of having high sequence identities, present very low coverages, as in Calipipla squamata, Pavocristatus, and Sirmaticus mikado, for example. On the other hand, we found probable occurrences of XPD in sequences coming from 2 species of ctenomyforms, additionally to 1 species of struthoniforms. As you can see here, the ctenomyforms Notoprocta perdicaria and Ctenamus guttatus have both XPD canonical domains, with the latter having also an additional domain HBB. In turn, Struthiocamelus is missing only the N-terminal domain. So, considering the possible occurrence of XPD in these species of ctenomyforms and struthoniforms, we use their sequences as queries for BLASTX searches against the chicken proteome. However, we did not obtain a canonical XPD as well, but the DDX11 protein was retrieved in 2 of these searches. Taking this into account, we suggest that a progressive loss of XPD sequence may have occurred from the base of the bird phylogeny throughout the evolution of galliforms until chicken. So, what did we find about the occurrence of XPD, helicase, in galliform birds? Well, we suggest that the DDX11 helicase may be replacing XPD function in chicken. We also found that there are probable occurrences of XPD in only 3 of 20 species belonging to Cynamiforms and Strutioniforms. And we suppose that there might be occurred a progressive loss of the XPD sequence throughout the evolution of galliforms. Therefore, this work reinforces the importance of in silico studies to open perspectives for functional investigations. I would like to thank CAPES and CNPq for the funds and the International Society for Computational Biology for the opportunity of presenting my work. Thank you for your attention.